Morning viewer, welcome to our Glory Globe TV. Yes, breaking news. DNA, deoxyribonucleic acid, inside the nucleus is not packed as a rigid regular fiber. Linker histone H1 dynamically binds and loosely glues nucleosomes together, creating a dynamic fluid organization that can still support essential genome function. The human genome has a remarkable capacity for compaction. If all 47 human chromosomes were stretched end to end, they would collectively reach about 2 meters in length, but they are somehow arranged within a nucleus only, that is below 10 micrometers in diameter. To fit inside the nucleus, DNA is wrapped around histone proteins like thread around a spool to form nucleosomes. Viewers, nucleosomes are associated proteins then fold into chromatin, enabling the genome to be packaged while remaining functional. For decades, molecular biology textbooks described linker histone H1 as a key factor that organizes nucleosomes into regular, rigid, higher order structures, often depicted as a 30 nm chromatin fiber. However, accumulating evidence suggests that such highly ordered fibers are very rare in living cells, and this raises a fundamental question. How does H1 actually compact DNA and organize chromatin in vivo? A team led by Professor Kazuhiro Mahishima at Shoken Dia and the National Institute of Genetics, that is called Research Organization of Information Systems ROIS in Japan, and Professor Rosanna Colapardo Guevara at the University of Cambridge in the UK. They investigated how H1 binds to nucleosomes and shapes chromatin inside living human cells. By combining cells and nucleosomes imaging with a superregulations fluorescence microscopy and computational modeling, the researchers reveal a new physical picture of H1 functions rather than working as a static clamp. H1 behaves like a dynamic liquid-like glue that gently folds chromatin into a fluid organization that remains accessible to other molecules. Beyond this exclusive report.